There's a couple things I wanted to mention with regard to tissue culture before we jump into the last bit of this content. And, you know, talking about this idea when we were looking at it on Wednesday, that you have to have such a clean and sterile conditions. You have to be so careful. I just wanted to show you some examples of, you know, this is a common problem. So this is a publication that we published in 2021 looking at doing cannabis tissue culture, and you can you can see as you look at the figures here. Whoops, that's not what I wanted. So you can see the microbes. So all this fluffy white stuff should not be there. Figure you can see the different colors, different funky looking bacteria. It looks like root tissue almost, but it's all bacterial growth. And very colorful. Here's like a greenish gray fungus. Here's a beautiful pink colored fungus, but again, not what you want to see in your tissue culture vessel. And so this is what we're trying to avoid. Now, I think I did mention on Wednesday, sometimes this is unavoidable. So all of these that you're seeing in the picture, when they studied them and looked at them, they were endophytes. So they were microbes coming out from the tissue. And in this paper, I put the actual article in the notes section if you want to look at it further. But in this paper, they tried different antibiotics to add into the media to stop this from happening, and they had very limited success. And so again, you know, it's not a guarantee that even if you add in antibiotics, you're going to get it to work. And you can see, at least initially, some of these plants, like I guess a figure F here, where you have the plant looking, you know, even this plant looks still not too sad. It's still green over here. It's definitely looking happy. But if you look at the last row here, you clearly see that the contaminant overwhelmed the system and started to, you know, essentially attack the plant and use the plant for nutrition. And so that's obviously not what you want happening. But you know, I've had many cases where you see the plant looking fairly happy, even though there's evidence of contaminants, and it stays in balance. So it just depends. Now, you know, you still have issues of if you want to try to propagate that or transfer it into a new vessel, you might still have contaminants that come with it. And so part of their research was looking at how do we minimize this whole contamination issue, and they tried options with very limited success. Here's another nice demonstration of contaminants, a couple of different kinds of contaminant ideas here, where the tissue, the plant tissue, it looks like almost like carrot tissue. I didn't look to see the details of this, but you can see the squarish pieces of orangey tissue. And then you can see the contaminants, and if you look carefully, you can see how the contaminants seem to be growing out from the tissue. Like this one here where my laser pointer is, this big orange blob coming out is bacteria. So when you see that pattern where you see it looks like it's actually growing out of the tissue onto the agar plate, that's likely an endophyte. However, these other colonies of microbes that you see scattered across the plate away from the tissue, that is probably a contaminant coming from the environment. As they're running the experiment, either came off the person's body. Hopefully they didn't sneeze into the plate, but you know, this type of idea where it's coming from the environment, then you tend to see the colonies growing away from the tissue. So that's something you should always key in on and think about, because that will make a difference on how you know, is my technique good or not? There's not a whole lot you can do about the endophyte issue, though, unless you hit upon a good antibiotic mixture. So I wanted to include those. I'll update the slide set to include these, so you'll have these images as well. Okay, so we've been talking about tissue culture and then other methods to propagate or genetically modify plants. The last topic we looked at on Wednesday, and I wanted to mention, I think we left off here pretty much looking at this idea that you can genetically modify plants for a variety of reasons. This is actually looking at poor quality soil if it has a lot of salt. And there are lots of salty soils out there, whether it be egg soils that get over fertilized and salt start to accumulate in the egg soil to the point of it's too salty, and then they have to abandon the field for a while. Here you're looking at the top row of the A, B, C, D, E. The A is the lowest salt concentration, and then the E is the highest salt concentration. And you can see this gradual decline in plant health as the salt is getting more concentrated in the soil. And then the lower plants were all genetically modified to be able to deal with salt tolerance. They have a salt tolerance. And so you can see that the plants, no matter even in pot E with high concentrations of salt in soil, they still are doing just fine. And so these types of ideas where you have poor quality soil, they're trying to find solutions to that. Because when you look at egg soil around the world, when you talk to people who study this issue, we pretty much have used up the best soil conditions, the best plots of land. They're pretty much already in production. And then we have poorer quality, lower quality land that is not being implemented, but now they're like, okay, we got how many more billions of people to feed by 2050? So we got to figure this out. So one strategy is to go into the lower quality soil systems and try to grow crops, or whether it be for food or fiber or fuel or whatever. But again, they might have to do some tricks like this in order to have a successful harvest from that soil system. So things that I think you're going to hear more about in the next decade or so, this idea of transgenic plants to solve some of these problems. Certainly, I think we left off here with the idea of flavor saver. Normally, when they ship tomatoes across country, they, of course, if they're fully ripe when they're in shipment, the bruising that can happen, just the physical shaking of the tomatoes and the bumping around in the semi trucks or whatever train system. And so they're trying to find a way to minimize the spoilage as things are getting either stored or shipped. And so for soft-bodied fruits like this is a big problem when you start looking at the logistics of how do you ship these types of produce across the country safely, so that when they get to market, you actually can get money for that, not have to throw them away because they're spoiled. Here's an interesting one. Cute little mouse to get you thinking about using mice as a tool to create what are called plantibodies. So this is kind of funky. What they do is they have mice that they will get exposed to plant pathogens, and the mouse will create antibodies through their immune system against that plant pathogen. And then you isolate the genes that are coding for that antibody against the plant pathogen. And then you take that gene and you put it into a plant. And if the plant expresses it, then the plant will produce those antibodies that are specific to that plant pathogen. So you can think of it as the mouse, like it's just being used as a tool to figure out what gene is allowing for that particular antibody, and then you take that gene out and put it into the plant. And so they named them plantibodies because you're working with, often they'll actually do tissue culture of mouse cells. I don't know if they still use the entire live animal or if they just have totally switched over now to doing, because you can do tissue culture with animal cells, not just plant cells. It's a little different beast, but you can do that. And you basically what you do is you just keep the cells alive and running. They don't like morph into a new mouse, for example, like plants can do. But very often cells in a tissue culture system so that you can study some of the biochemistry and physiology stuff. Anyway, so plant antibodies is the idea of getting the antibodies expressed, figuring out which gene codes for that antibody and getting that gene to the plant. And they've done this to completion where they can show that it works. The plant will pick up that stretch of DNA, that gene, through tissue culture techniques, grow up the plant, and then expose it to the plant pathogen, and they will express that gene and get the antibody against often it's some kind of toxin that the pathogen's producing. They'll be able to inactivate it. So really kind of a funky idea, but it works. So, when you start looking at options like, what could they do? What could they use this for? There's a lot of options. I'm listing out a few here, but there's more out there. One of the first ideas was, can we create disease resistance in certain crop plants? And so there have been, since, crops have been domesticated and grown for thousands of years, there's been pathogens that come along with that system and can wipe out entire crops. And so this is nothing new, but to be able to genetically modify the plants to better deal with getting attacked by pathogens is a big deal. This is a huge area of effort. And again, fungi tend to be the number one culprit. There's more fungal pathogens out there that attack plants than bacterial pathogens. And then they're looking at, okay, if we have to feed all these more people, but we have limited land, it's not like we have a bunch of land we can keep, growing more and more crops. We're pretty much maxed out. So another strategy would be make sure that each thing that you harvest has greater nutrition per gram of tissue. So make a better tomato that's more dense in its nutrition, or corn on the cob, where you look at the kernels and they're more dense in nutrition. And so this is another thing that they're looking at for the use of genetic modification, increasing the yield, just the overall yield of whatever part of the plant you're trying to harvest and get out into the market. They've done that with rice. So one of the things they're really working on is we're going to learn a little bit in that plants can make sugars a little differently from each other. Some plants can do it very efficiently, and we call those C4 plants. Rice is not one of them, but rice is, $1 billion crop. It feeds so many people on this earth. And so the idea there is if they could take genes from plants that do this special C4 process, stick it into the rice plant and have the rice plant express that. This is a big endeavor because when you look at the process of how these so-called C4 plants do their thing, it's not straightforward. It's a multi-step process, physiological pathway. So it's quite daunting. I give the researchers a lot of credit for going after this. And if they can get this to succeed, they'll probably win the Nobel Prize, would be my guess, because this is a big, big accomplishment if they can get it to work. Anti-cancer, of course, just directly for human health. They're looking at drug, being able to use the plants as drug factories, essentially, getting the plant to make certain drugs. Edible vaccines, I'm not sure. I think this is still being looked at. And surprisingly, I was surprised to find out that the white potato is being used as edible vaccines. So they'll have the potato tuber express the necessary material that you would need to be vaccinated against certain diseases, like certain viral diseases. So the reason they're doing this is when you go to third world countries and you have to vaccinate all these people in these tiny villages, they don't have like refrigerators for vaccines to stay cold and viable, and so they have to come up with another way. How do we vaccinate all these people when just getting to those areas and actually, sticking a needle in them and vaccinating them, it's not going to work so well. And so They said, well, maybe they can grow their own vaccines. Give them this potato plant and have them grow that up, and then they can eat the tuber. And so I've heard about this now since I was a grad student. They're looking at this, still trying to get this to happen. A lot of third world countries in particular, we're pretty fortunate here in America, but in many other countries, there's key vitamins and minerals that people are lacking in their diet because their diet isn't diverse enough and they can't afford the diverse diet. And so this is a huge area of research. When I went to a plant conference right before COVID in 2019, there were big symposium sessions just on this topic, like how do we ramp up vitamin and minerals in plants? And one way they were looking at doing that through transgenic processes. So there has been success. They've done iron into rice, so they've ramped up iron in the rice grain, and they've ramped up the protein levels, which is big because rice doesn't have a ton of protein in it. So if you can ramp that up, again, especially in third world countries, it's a huge bonus for them. Vitamin A, you've heard of golden rice. Vitamin A is what is being ramped up, so the rice grains actually look golden. That's why they call it golden rice. And so the salt tolerance idea that I told you about, where there's just a lot of soil out there that's too salty for normal plants to grow in. And then I'm seeing a lot of published studies now on this idea of using plants to clean up toxins. So that process is called phytoremediation. Phyto is plant, remediate is to clean up. And so they're looking at modifying plants in order to better clean up so many of the toxins that we've introduced into our environment through our human activities. Okay, so this is just a partial list of ideas of why scientists are interested in this idea of introducing genes to plants. And, working with plants, because they're so versatile, and especially with the agrobacterium model, that method of getting genes into plants is pretty easy. So it's really kind of opened up the field of possibilities of what we can do in order to improve human health or the environment. Here's the golden rice idea, very obvious difference, just visually. Okay, so the introducing different genes in plants, there's some other tools, I guess, I'd like to explain that are being used in commercial crop production. So if you have two plants and you're saying, well, I like this plant because it has this quality or characteristic, and I like this plant because it has a different quality and characteristic. Wouldn't it be cool if we could just combine that into one plant? So this is this idea where you can't get them to breed. They're sexually incompatible because they're different species. So we can't breed them. So then what? And so this idea is pretty amazing. It's this idea of fusing the protoplast. So again, in tissue culture techniques, you work in the lab using tissue culture techniques, you grow up the cells of each of these plant species in one container. You strip off the cell wall. Remember, that's what a protoplast is. It's just a plant cell that has the cell wall stripped off. And so you have these different protoplasts swimming around in the same broth, liquid, and then you get those to fuse. So if those cell membranes fuse with each other, often they apply like an electric current or some kind of chemical like the polyethylene glycol, for example. You'll get those protoplasts to actually fuse with each other. And then you allow, through tissue culture technique, you encourage that to happen, and then you encourage that fused cell to divide, grow until it becomes a plant. And hopefully you'll have the characteristics that you want in one plant. And it sounds really bizarre, but they call it hybrid fusion. That's kind of the process that you're looking at, because you're using two different plants. So you're creating a hybrid from these two different plants. So, I mean, plants are amazing that you can get this to happen, and you'll actually end up with a living plant at the end of this. It won't, die off. So it's pretty amazing. So then you might see the term somatic hybrid. That is often, I see that in the literature. They'll say it's a somatic hybrid. And that just means that somatic means just regular plant cells, not the sperm and egg. So you have gamete cells, and then you have somatic cells. Like most of your body is somatic cells, and then just your sperm and egg are the gametes. So when you see that word somatic, that means they didn't use sperm and egg to get this to happen. They took just plant tissue cells and merged them through protoplast fusion. So this is a interesting example. The plants on either side have different resistance to certain heavy metals. And so the one on the left, I have in the notes section, that's Indian mustard on the left and stonecress on the right. And then the hybrid is in the middle. And so the one on the left, the Indian mustard can tolerate lead, but not zinc or nickel. But then the one on the right can tolerate zinc and nickel, but not lead. And so if you take these and do protoplast fusion and get this plant to grow up, it's resistant to all three: zinc, lead, and nickel. And you can see it's quite happy. It's growing quite well in that soil. And so these types of ideas, and they just took, normal plant cells. They weren't working with sperm and egg of the plant cells. So you might see this term as well, somatic embryo. So again, what they're saying is this is an embryo created through somatic cells, not sperm and egg, which is weird. But what they do is they can get, through tissue culture technique, they can get a cluster of cells to form that behaves like an embryo, even though that cluster cell, it did not come from sperm and egg fusion. That's not what's going on here. And so you can, through very special tissue culture techniques, get this cluster of cells to form, and it starts to behave like a plant embryo, a developing embryo. And then what you can do, you'll encourage that to keep growing and growing until you get essentially a mature embryo. And then what you do is you encase it in a protective gelatinous matrix. And then they sell that. They call them artificial seeds. So you might see that term, if you go to buy certain seeds, they might refer to them as artificial seeds. So it's this really bizarre process where you can get body cells of the plant to behave like embryo cells. And then you just encase all of that in that matrix and you can sell it off. So this is becoming a little bit more mainstream. It's took a while to, there's a lot of work that goes into this to get it to work successfully. But I'm starting to see more of these terms out in the real world now. So you'll probably see more and more. So these are all strategies that can be employed to try to deal with, creating plants to have certain traits, desired traits, even though they're not sexually compatible with each other. So you can't do it through normal breeding methods, but you can use a bunch of these other methods that we were just talking about to get it to happen. So in lab, hopefully next week, you're going to be working with Agrobacterium tumefaciens, and you'll be trying to genetically modify Arabidopsis that you've been growing. So we're going to take a stab at that and see how it works in lab next week. So that's one of the things you'll be doing. And then we're also working on getting tomato seed sterilized and growing in tissue culture medium to the point where then they'll create little plantlets that we can give to you, and then you'll take a stab at doing tissue culture with those. So that's going to probably be in a couple weeks out yet, because we have to let the tomato seeds grow for a bit. Okay, so biotechnology, some really amazing advances that are going on in that general field. And then another field that's kind of up and coming now is called synthetic biology. And so that is a lot of kind of many of these ideas where they're tinkering with physiological pathways through genetic modification. And they do use bacteria and fungi for this, like yeast especially, but they're also using plants for this too. When I went to that plant biology conference, there's like a whole symposium session on synthetic, they call it synthetic biology. So sometimes they shorten it, they call it SynBio for synthetic biology. So that's just something that's with the technological advances that we have now. When I sat in that session, there was like a whole day session, and my mind was just getting blown left and right because it was amazing what they were able to do with the technology they have on hand. It just the complexity of it was just mind blowing to me. Okay, kind of related to this topic, I just posted this slide set in EduCat just a minute ago. Related to this idea of tissue culture techniques and getting plants grown up the way you want them. I wanted to mention propagation. I know that those of you in lab have taken some stem cuttings primarily, probably, and are trying to propagate some plants that you had available to you in the greenhouse. So I wanted to mention a few things about that, because propagation is huge in commercial production of plants. Propagation is a very common, pretty straightforward method to create many, many copies of the plant that you're interested in getting many copies of. And so primarily what you did in lab was you probably took stem cuttings, but basically, again, because plants are so amazing in their ability to, you can take a stem cutting and you can still encourage that plant to you really don't have to do a whole lot other than put the cut stem into some water or even directly into soil, and it'll start forming roots, even though you just amputated it off some other plant. It can still go, oh, all right, well, okay, I don't have any roots anymore. I guess I better grow my own. And so this idea that pretty much any part of the plant body, you can take off the mother plant, take some kind of cutting, and then if you know some basic skills, you can get that to grow into a complete plant again. And so you have to be a little careful about providing the right environmental conditions, but it's not terribly difficult if you just know a few things. And it really is dependent on the type of plant, what kind of propagation technique you would use. And so there's all kinds of options. Stem is probably the most common, but you could just take leaf tissue, like cut off a leaf and plant it. I've done that with ZZ plants. Actually, I had a student do that for me, and you have to be patient, but now I have ZZ plants growing just from a leaf that was taken from the mother plant and put into soil. I kept the soil moist, and it's been about 9 months now, and I have baby ZZ plants coming up. So you can do just leaves. You can just work with the leaf bud. It's not even quite there yet. It's just the bud stage. You can work with just the roots. So again, plants are just so amazing in their ability to essentially, even though you've amputated them, they can grow, they can figure out essentially what's missing and regrow it. And so this is just looking at the basic ideas of different types of propagation. So I'm just going to say a few things about some of these. So for stem cuttings, which is probably what most of you did in lab, you're taking, the above ground portion of plant material, focusing in on the stems, and you can actually take multiple stem cuttings from one stem, as long as you have, especially if you make sure that there's an axillary bud present, then you'll get very quick growth, typically coming from the axillary bud meristematic tissue. And so typically you take a cutting, you remove some of the lower leaves, and then in lab, depending on how you chose to do it, you could either put that cut stem into water, and I like doing that because then you can watch the roots grow, or you can actually just put the stem into soil and just make sure that the soil is watered, but then you can't watch the roots. So basically, what you're doing when you take a stem cutting, what's so amazing is that essentially the plant goes through this process called de-differentiation. So right at the cut area of the stem, those stem cells there have to de-differentiate. So they have to go, oh, okay, well, I was a stem cell, but now that I've been amputated off the plant, I need to do roots in this section. And so they'll stop being the stem tissue cells, and then they will shift into root cell growth. And so roots will start to grow from there. And so this de-differentiate process is amazing. You know, like there's a whole bunch of shifting in gene expression to get the right genes turned on to take what was stem cells growing. Now they got to morph and start growing a whole new root system. And so it's an amazing process. Obviously, you got to be careful how many propagations you take off one mother plant. You can't go too crazy with it, but you can actually cut down a plant pretty dramatically, more than what most people think, I think, and still have that survive. If you're going to do this mass production of it, you typically see this kind of setup in a greenhouse where they have the cuttings. It's, again, stem cuttings are pretty popular, and you will have pretty loose soil substrate of some sort, and sometimes even soil. Sometimes they just go with some kind of like peat moss vermiculite mix. So it just depends on their production methods. And you tend to have misting systems in place to provide some nice humidity and moisture. And you don't go too crazy with intense sunlight or light, I should say, artificial light either. And you tend to baby the root zone. So often they'll have heat mats that you'll have the trays sitting on to encourage, when you're trying to sell these for production, for profit, you want this to happen quickly. And so you try to optimize all the conditions to promote really quick growth and differentiation into roots so that you get really healthy, robust root systems. So here you're seeing some stems that have formed the roots now, and again, it's this idea where those stem cells at the base of the stem that you cut are all having to morph into the root and keep growing and make a root system. And as that process is occurring, you tend to shift things. If you keep the humidity too high for too long, they tend to see issues pop up with the root system growth, and you get fungal infestations. And so you start to gradually stop babying them so much with the humidity and mist, and then you can also start ramping up the light scheme. And so that when they have the goal is as soon as they have a nice, robust root system, they're good to be transplanted and then, sold off to market. So again, they try to optimize it to make it go as quickly as possible, but it does depend. It depends highly on what plant species you're working with. It can be just a few days to get root systems to form, but it can also be several weeks depending on. So it's a very, they know a lot of the parameters for all these different commercial plants that they do stem cuttings with. So it's kind of just following the recipes. They've figured out a lot of this quite well. Here you're seeing a tray of, this is Streptocarpus leaves, which I have one actually at home. And This is a plant that's related to African violet. This is a very common house plant. It's very popular because similar to African violet, it flowers continually, like year round. You always have beautiful flowers coming off the plant. And so that's one reason I love African violets and their relatives, because they're always in bloom. But here, what they are doing is they took leaves, again, just the leaf, and they put them into soil, and you can see the baby leaves or the baby plant coming off of it. So like this is what they put into the soil, and then the babies are sprouting up right there. Okay. So just taking a cutting of the leaf. And so you can get quite a good production method going with that. And you can do the leaf blade. So I put this inset picture just to remind you of the terminology here. The broad part of the leaf is called the blade, and then the stem is called the petiole. That's what attaches to the main stem tissue. Okay. And then again, remember the node region is where you have the leaves attaching and where the axillary buds are located. And so node tissue is very popular to take, as making sure that you can have node tissue and axillary buds there, and then you'll get good propagation through that. They also use node tissue for tissue culture because that's usually a very active area of growth. It's meristematic tissue there, so that's a very prime part of the plant that likes to just divide, grow, grow, grow. So you can do leaf cuttings, like you can cut up the leaf essentially and get new leaves to form, or new plants to form from that. And so you can take one leaf and get actually many plantlets from that. As long as you know what you're doing, you can get multiple plantlets coming off one leaf that you work with and propagate. And so again, the leaf blade, you can have this happen. You can use the petiole tissue or the axillary bud tissue, and or all three. Sometimes you just make sure that your stem cutting has all three, and you're good. So when we teach propagation, that's typically what we do. We ask you to take a section of the stem that has leaves, petiole, and axillary buds, because that almost guarantees success for getting good root formation and a healthy propagated plant. So we tend to do it that way. But you could just work with root tissue if you wanted to. And it's kind of bizarre, but it works. You take the roots and you get them planted. You have to make sure that you're keeping a tight eye on the soil. Of course, if you're doing this commercially, it's pretty much automated in greenhouses where they have the misting system all computer controlled. And then you get, from that, you'll get adventitious stems popping up out of the soil. And of course, they'll form the leaves and all of that. So you end up with a complete plant. And so, again, they have these protocols out there, and they've been able to troubleshoot through these processes. And it depends on the plant species, which part of the plant you're going to pick in order to do these propagations to get your plantlets to grow. And certainly anytime you're working like with your stem cuttings that you currently did in lab, age of the plant does come into it to some extent. And so, if typically you don't want it too young of a plant, but you don't want it too old of a plant either. If it's in a vigorous phase of its life cycle, that's usually good success. The time of the year, so if you are trying to work with herbaceous plants, it's usually not too big of a deal as long as you have healthy plants. But if you're working with woody perennial plants, so a lot of plants that are used for landscaping, things like that, you got to be careful when you do this kind of propagation because they have a more fixed life cycle that can be challenging to figure out how to work with them if they're in a phase of their life where they're starting to go dormant, for example. Then trying to do this as they're shifting into dormancy, it's really challenging, if not impossible. So You got to be careful. I still have not tracked down why, like what is really going on here, but I've seen this in several different resources. They say etiolated seedlings can be easier for root development. And so I don't know the actual reason for that. I'm still trying to chase that down. But this picture that you're staring at, the plants on the left are normal, it's like bean plants. The ones on the right are etiolated. So I'm sure you can easily see how the stem tissue, they're much taller plants, and then the leaves look pathetically kind of yellow, the stem's all white. That is an etiolated plant. So how does it do that? You can do that if you want, but it looks pretty sad. You take like seeds and you germinate them in the dark, and then you keep the seedling in the dark. So, like in was it the first week of lab? Didn't you do the paper towel method where you got the seeds going in wet paper towel, and then we put them in the cupboard? Well, if you just plant them in soil and put them back in the cupboard where they don't get any light, this is what they do. And so it's called etiolation. Basically, that plant is going, oh, I'm in the dark. I'm probably getting shaded out by a neighbor plant. So I'm going to grow tall to get above the canopy so I can get access to light. So that's why they shoot up in height. And because they're in the dark, they don't have any major chlorophyll maturation going on, so they don't look green. But the good news is, if you take that etiolated plant and now put it under lights, it will survive. It'll green up, it'll start to behave more normally, grow more normally. Anyways, for some reason, etiolated plants, if you do that to plants, get them in the etiolated state, they can be easier for getting the root development going. So you can certainly fuss with hormones. Often auxins are applied to get rooting. So you might have done that in lab where we had rooting powder in the fume hood that you could have dipped your cutting into if you wanted to. And that powder contains auxin hormones that will stimulate those cells right at the cut region of the stem to start forming roots. So that's a very, very common strategy. And then misting, making sure that you're being careful about the humidity and water levels. They find that misting works really well to get good rooting to happen. And again, warming up the soil, they find it really does help. So if you know a few tricks and ways to really optimize the rooting of these different stem cuttings, it works well and works quickly. Here's some interesting ideas that work depending on the growth form of the plant. Tip layering, literally taking the tip of the shoot and burying it. That's all you got to do. And then you can see that it starts to root and grow a new shoot. And then simple layering down below, you just kind of curve it and again, anchor it into the soil. And where it's in the soil, it starts to form the root tissue. And then you just cut that free from the mother plant. And you have a little plantlet to work with. So it's amazing how easily that these plants will respond to this. Mound layering or air layering. Mound layering, you just take the soil around the plant and mound it up onto the stem tissue. So some of the stem tissue is buried, and they'll start forming adventitious roots right off the stem. So then you can cut those, and so from that you have 3 little plantlets that you can take and put into new containers. Air layering is a little bit more work because you have to kind of cut the stem tissue, and then you tend to wrap it with like sphagnum peat moss and then some kind of wrap around that so the peat moss will stay humid and moist, and then you get roots forming inside there. So you have to have patience. I've done that with some plants. You got to have some patience because you have to kind of like carefully unwrap it and look to see like anything happening yet. Usually it's like, nope, got to rewrap it and keep it going. So air layering can be a little bit trickier. But it's amazing how, you can just do these little tricks and you get these little plantlets that will form off the mother plant. So strawberries are really easy to take some of the runners or stolons and just bury those. And again, it's kind of like the tip layering idea. So-called suckers, you see that word a lot in the common literature. It's just plants that come up off of horizontal roots that are growing across the surface. The raspberries, blackberry plants are often propagated through this method. And then sometimes you have to take the whole plant. You might see the phrase crown division. That's where you take essentially the whole plant. Hostas are done this way. Hostas are big business. There's a lot of hostas for landscaping. And so you tend to take, when they say divide the whole plant, that's literally what you do. You take the whole plant, get it out of the soil, and you cut it in half. That's usually what I do, and plant the 2 halves all the way through to, half the root tissue as well. So, I mean, you can be pretty dramatic in hacking and cutting off different plant parts and then encouraging them to grow into complete plants. So then you have these specialized portions of different plants, the bulbs, corms, and tubers. So you're looking at a potato tuber there in the picture, and you can see those, the eyes of the potato are really the node tissue. And so from the node areas, you're getting new stem and leaf production. So if you were to take that tuber and plant it into the soil, you would definitely get an entire plant from that. And so when by the way, it's weird because if you want to buy potatoes for your garden, if you want to grow potato plants, like I want to grow potato plants, what you end up buying, and I remember the first time I heard this, I was so confused. They call them seed potatoes. And I'm like, wait, what? Seed potato, what do they mean by that? And then they'll sell you a bag of little potatoes, the tubers, like the stuff you'd eat. And it's like, well, that's not a seed. What do they mean? But basically what they're trying to get across is that as long as they give you a tuber that has these nodes and you plant that tuber into the soil, you'll get a full plant. It'll form a full potato plant. And then you let it grow for a while, and at the end of the season, you'll have lovely potatoes to dig up and eat. And so seed potatoes commercially, they're also certified to be disease-free. And so that's why they kind of give them a special name, because if there's true seed potatoes, they've been guaranteed to be seed-free, pathogen-free. And then, of course, you can use rhizomes tissue, which is underground stem. Okay, the last topic I'll talk about, but we'll save it, is grafting. I want to talk a little bit about that because that's becoming more and more common, primarily because they're figuring out, the tricks to how to get this to happen more successfully. So we'll talk more about that on Monday. We gotta stop.